All right, guys, happy Friday. It's Miss Hall here. This is kind of the big picture of what we're going to cover today. You should have already gotten out your day 9 notes. We're not moving on to day 10 just yet because we've got to figure out what is actually happening. Yesterday, can you go grab the characters on my desk? Yesterday we went through our cast of characters and we talked about who all is going to be involved in this whole shindig here. I'm going to flash that back at you. This is a colorful picture, not like the one you drew yesterday. You probably drew it in pencil. So if we remember, our innate immunity is run by the macrophage. He's going to go through phagocytosis to engulf any of our pathogens. We know that when the pathogen is actually presented in our skin, we have mast cells that are going to trigger the inflammatory response. Think in your head right now. We'll do a little Dora the Explorer. What are the 4 things that happen in the inflammatory response? Okay, think. Ready, and the first time, they're going to turn red, all right? Things are going to turn red when they're inflamed. They're also going to get, you said it, warm. Very good. They're going to get warm. We're also going to have some swelling in the area. And what am I missing? Red, warm, swollen, painful. Duh. Okay, painful. They're going to get painful. So hopefully you thought of those 4. I had trouble thinking of that last one, so maybe I need to go back and study a little bit more. So the macrophages will show up, and they will start to engulf those pathogens, and they are going to do one specific thing to help everybody else look out for that same guy. That's right. They're going to smear his guts all over their cell membrane so that they can show off the guy they just killed. All right. Now, that's the innate immunity. That's the immunity you're born with. But we've got all these other characters that we've got to talk about as well. So we're going to zoom in on the lymphocytes. The lymphocytes are the ones that are going to run what's called our specific immune response. The other 2 are our innate immune response. We don't have to work on them at all. They're going to show up just by being human. But these specific immune responses are going to have to be kind of taught how to do their job. And we kind of have to learn what our environment is like before we start getting these specific immune responses. Yesterday, you learned that we have certain cells called B lymphocytes. These B lymphocytes are going to be circulating within our fluid. We call those our humoral response. Our humoral means fluid. So these cells are going to be circulating in your blood, circulating in your lymph system. That's your lymph nodes that you hear about getting swollen sometimes. If your lymph nodes are swollen, that means that these B lymphocytes are actually going to work. And then we also have our cell-mediated response that's represented by all the red type of cells. The first one that's going to be initiated is our T helper cell. They're going to be the ones that actually tell the B cells to get to work and that are actually going to activate our T cells. Our T cells are kind of mean guys. They're like the big bouncers of the club. They're the ones that the mob hires to go do the job. So they are actually going to attack all of the infected cells that are in our system, the cells that the virus or the pathogen has already taken over. And they actually can do a little reconnaissance and search out those cancerous cells that may be in our body. So they can recognize the mutated proteins and the proteins that aren't quite acting correctly in those cancerous cells and take care of them for us too. And we know they're going to use this chemical called perforin. That chemical is kind of like little cellular bullets, and those bullets are going to poke holes in the membrane and allow fluids and ions to exchange sides and cause different reactions to occur that make that cell swell, and the cell will eventually explode. We also have some natural killer cells, and these cells are going to be used once our antibodies have kind of jumped to action, and they're going to be able to locate the cells where the antibodies have kind of bunched up and tagged those bad cells, and those natural killer cells will come in and take care of those pathogens that have been attacked by antibodies. All right, if you're unsure about an antibody, we kind of covered this in some of the classes that we had time for, and some of them we didn't. I'll leave this on here so you can see it. Antibodies are kind of Y-shaped, kind of a double-legged Y. They are also called immunoglobulins. That's kind of hard to see, but it's this word right here. Immunoglobulins, and you'll see where they get that name as soon as I explain to you how they work. They are made and secreted by our B cells. Our B cells we know are going to mature into two different types, plasma cells and memory cells. The plasma cells are the ones that are actually going to make those antibodies. The first time we're exposed to that antigen, they will start cranking out those antibodies. Does anyone know how fast they'll do it? Dora time, 3, 2, 1. That's correct, 2,000 per second. All right, hopefully you were right. And then for later, when we actually get over our sickness, we may end up being exposed to this again, especially if we're kissing boys or shaking hands or not washing our hands after the potty or whatever. Maybe we went to a concert and we're around a bunch of people or got on an airplane where we're all circulating the same air. Those memory cells are going to stick around for when we actually get exposed a second time to that very same antigen, and those cells will start to jump into action and start cranking out antibodies to be able to fight it the second time around. All right, now the things on our antibodies that we really need to know about, we've got a variable region represented here by that gray color, and we've got a constant region. What this does is this variable region is the portion that's actually going to attach to the antigen, and then the constant region is the portion that our macrophages, zooming out, our macrophages will recognize, and they'll be able to attach to that constant region and start to the engulfing process. So kind of a cool concept. All right, so now that we've got our cast of players, let's go back to the drawing board and figure out what the heck is happening in the immune system and how all of this comes together. Well, let's start at the beginning. You got to have a pathogen. So let's say that you licked your shoe or someone sneezed in your face. Happens a lot with baby Charlie. Maybe you, like I said, got on an airplane and someone else was breathing some gross air out and you happened to breathe it in. All along the outside of the pathogen's cell wall or cell membrane is going to be these little receptor proteins. These receptor proteins are referred to as the antigen. It's the antibody generator. So if we didn't have antigens, there'd be no need for antibodies. So this antigen is present in plethoras on the surface of that pathogen. Now, this pathogen might be, hopefully this isn't as blurry as it looks on the camera, might be a virus, or it might be a bacterium, or it could even be a microbe, just a small microorganism that's trying to make you sick. We call this pathogen the bad guy because he's the one that's going to set this story in motion. He's the one that's going to cause all the problems. If he didn't show up, everybody'd be happy and we'd just be running like normal. All right, so the first path we could take once the pathogen is present in our system is we could go over here and infect a cell. So if the pathogen is undetected right away, it could be floating around and it might infect a red blood cell or it might infect a skin cell. The funny thing about pathogens is they are usually very specific as to which type of cells they're going to infect. So let's say we're the flu virus. If you're the flu virus, what cells do you think you're going to infect? Hopefully you said throat cells. Very good. They will infect throat cells. Now, if you are some other type of pathogen, whatever you are going to be specific to a certain number of cells. If you remember yesterday, I told some of the classes HIV. HIV is definitely a pathogen. So what type of cells is HIV going to infect? Hopefully you remember, it's the helper T cell, and that's going to be a big problem once you see what the helper T cell is responsible for. All right, so first pathway is maybe that pathogen infected a cell in our body. Well, that cell has really no defense mechanism just on its own. It's going to just get taken over, and that pathogen is going to jump in there and start hijacking the cell's machinery, and it's going to start making copies of itself. What this cell does in its last dying moments is it will take a piece of that pathogen, the antigen specifically, and it will place it on its membrane. You can see it here. Once that antigen is placed on the membrane, we now call this an MHC1. That stands for Major Histocompatibility Complex. That's a big fancy word. I'm fine with you hearing or saying MHC. So that MHC1 is going to be kind of the signal to other cells in the immune system that this is the guy we're looking for. So they're trying to sabotage, that infected cell is trying to sabotage the war that the pathogen is waging on us. Now you'll notice this infected cell also has a little bit more revenge that it can cause. It will release the chemicals called interferons. Interferons are like the cell caution tape, if you remember from Wednesday's lecture. That cell caution tape is going to use negative chemotaxis. If you remember, chemotaxis means chemical movement, and negative means it will cause those cells to move away from the infected cell. So it kind of sends out this signal through those interferons that, hey, there is a bad guy in the area. He already got me. Try and save yourself and get away from me. Kind of nice of that infected cell. Now, another idea is that as the pathogen enters the system, maybe the first guy it runs into is a macrophage. If we break down that name macrophage, that means big eater. We know that it eats through phagocytosis. It may eat this pathogen if it sees him first, and it's going to take that antigen from the surface of the pathogen and place it on the outside of its cell, kind of like a trophy to say, hey, look what I caught. I'm going to smear his guts all over the outside so everybody else knows what he looks like too, in case his buddies run into the same kind of guy. When this antigen is placed on the surface of a macrophage, it's called an MHC2. Anybody remember what MHC stands for? Hopefully you talked to the screen and said Major Histocompatibility Complex 2. At the same time as it's digesting this pathogen and making sure that it's got the antigen placed on its skin, it's going to release a chemical called interleukin-1. We can abbreviate that with IL-1. What that does is that's going to use positive chemotaxis. Positive chemotaxis, that means movement towards the chemical being released. And that positive chemotaxis is going to stimulate the helper T cell. Okay, so far we've got the pathogen that can infect a regular cell, the pathogen running into the macrophage, and this will set in motion another step. So this helper T cell, it's going to move through positive chemotaxis due to that IL-1, that interleukin-1, and it's going to take some receptors on its surface and it's going to analyze that antigen that's being displayed on the surface, and it's going to create a little analyzing protein. There is a fancy name for this, but I don't need you to know it. So that analysis of MHC2 will tell it, okay, yes, this is a bad guy. I need to release my chemical to set my cascade in motion. So he's going to release IL-2, that's interleukin-2. Interleukin-2 is going to set some stuff in motion. But just to recall, we're going to read and display that analysis protein on the surface in order to communicate what antigen we're actually looking for. So we've got to place that analysis protein out here before we can set this in motion, because these new guys on these new pathways won't know what we're looking for if we don't show them. Okay, we've got to communicate. They can't talk to each other. They've got to show either through chemical or protein analysis. All right, so here's where we get to the tricky part. There's two things that happen. The first thing that's going to happen is we've got to stop the pathogen from infecting cells. That's idea number one. So in order to do that, we're going to go into our humors, or start our humoral response. Tell me again Tell me again what the humors are of our body. Anyone remember? Hopefully someone yelled out fluid, okay? It's the fluid of our body. So things in our blood, in our lymph system, we're going to go search out all those antigens. So the B cells are the first thing that's going to be activated. These B cells are going to, whoa, that's messy. Hopefully you can read that. We'll have to see later. They're going to read that analysis protein from the helper T cell, and through positive chemotaxis, they'll be pulled towards that IL-2 that's being released from the T cell. So the IL-2 calls them in. They're going to analyze that protein that's on the surface of the helper T. It kind of like made a cast of the antigen. What that will do is that's going to activate maturation. Okay, so this B cell is now going to mature into a couple different cells. Now, how are we going to turn one cell into two different cells? Any ideas? Maybe it was a topic we just recently covered. That's right, mitosis. So through mitosis, yes, these cells are going to have the exact same DNA present, only some of them will end up being plasma cells, and some of them will end up being, what is it? Memory cells. Very good job. So let's talk about plasma cells first, because these are the ones that we're actually going to use the first time we're sick. So through maturation of B cells, we end up with plasma cells. These plasma cells are going to create and secrete the specific antibody. This antibody is going to match perfectly with the antigen that was originally on the pathogen that got eaten by the macrophage, that got displayed and read by the helper T, that got analyzed and then passed on to the B cell. Okay, so here's our pathway. Pathogen, macrophage, T helper cell, B cell matures into a plasma cell. So these antibodies are going to match perfectly. And it's kind of cool how antibodies work. They're kind of like, I like to call them, excuse me.
Please send seniors to the gym. Seniors only to the gym, please.
Seniors, don't go anywhere. You're probably not here anyway. These antibodies are actually going to bind to the pathogen when they come across them. So let me switch out some pages real quick, and I'll show you how this works. All right, so the pathogen, there we go, has this little antigen on its surface. And there may be 2 or 3 pathogens in a single area. The antibody is going to walk up to these pathogens, and the Y shape of this antibody will bind with that antigen. And so this one may get another Y shape, and this one may get another Y shape. And eventually you'll see that due to the fact that there's 2 attachment sites on every antigen, we end up getting kind of a glob of pathogens all in one place. Hence the name immunoglobulin. So they kind of muck up the plans of the pathogens by kind of bunching them all together. Now you say, well, Miss Hall, why'd you call them toddlers? Well, I kind of imagine antibodies as like the little kids at Chuck E. Cheese. And when the mouse comes out, they usually just kind of go maul the crap out of the mouse. Okay, they're going to jump all over them. So it's the same thing that happens with antibodies. When the antibodies are presented with the specific antigen that they match, they're going to go nuts, and they're going to attach anywhere they possibly can. And then they're going to attach to any other pathogen they possibly can. So we end up getting a huge clump of these pathogens all in one place. Now, why on earth would we want to clump up all of our pathogens? Who's coming behind us to pick them up? Sorry, time out.
At this time, teachers, we need all juniors headed to the lobby. The gym. At this time, we need juniors headed walk.
Okay, all right, glad that's over. The white blood cells, or the macrophages, remember, are going to read this constant region. And so this is like a little flag to tell everybody else in the body, hey, I found the bad guys. Come and grab them. Come and eat them up. So they're flagging all of these pathogens to tell the body, come and collect them. Come and eat your snack. We got all the bad guys in one place. So it's kind of nice. These antibodies aren't actually killing anything. They don't do anything wrong. They just go and attach where they need to attach. And they're gonna flag and make sure that everybody knows who to eat. So back to our list here. So that's our plasma cells. Our plasma cells are going to do the job of flagging all those bad guys so that we can make sure we dispose of them properly and get them out of the fluids and out from infecting any more cells. Now, in the meantime, we've also created memory cells. These memory cells are going to be stored in our lymph nodes. Our lymph nodes, you see some in your neck, you've got some under your armpits, and kind of the creases of your hips. You may notice that every once in a while they get swollen. You don't feel sick, but that's actually your body mounting an attack towards some kind of pathogen that's in your body. And if your immune system's doing great, then you usually don't even see any symptoms of it. But sometimes your immune system isn't quite on top of things as fast as the pathogen's able to replicate, and you do get a little sick. If you've ever had mono, which I definitely have, your lymph nodes get humongous.
Teachers, at this time, we need sophomores to the gym. Sophomores headed to the gym.
My lymph nodes were described as cottage cheese covered golf balls when you look down my throat. So that was really, really gross. My memory cells had not ever been exposed to mono before, so I didn't have my antigens built up yet. And so I had to wait for this whole process to click into motion. These antigens were antibodies were finally produced to start beating back that virus in my system. And so now I have the memory cells. So essentially now I can drink after anybody I want and kiss whoever, well, not kiss whoever I want because I'm married, but now I have an offensive if I ever get exposed to mono again, which being a high school teacher, I'm sure I have come in contact with it once or twice since having it when I was in high school. All right, so let's zoom back out. We had the pathogen get discovered by a macrophage, which turned on our T helper cells. Someone remind me, what's the chemical that's released? Time out.
Teachers, at this time, please allow freshmen to go to the gym. Freshmen. And anybody else that needs to go to the gym, please head that way at this time. Thank you.
I say, no, Miss Davis, I'm not going anywhere. All right, so the macrophage releases a chemical that tells the helper T cell to kick into gear. Anyone remember what it is? Good, interleukin 1, interleukin 1. And then the T helper cell is going to release a chemical that sets a couple other pathways in motion. Anyone remember what that is? Interleukin 2, very good. So interleukin 2 is going to turn on the B cells. We get the plasma cells and the memory cells from that. But there's also cells that have already been infected. Okay, the B cells are going to take care of the actual pathogen floating around, but what if the pathogen has already infected some cells in our body? Are we just going to let them keep replicating and keep passing that pathogen from cell to cell? Absolutely not. So we're going to take care of our fluids on this side, but we're going to take care of the cells that have already been infected by our cell-mediated response. The cell-mediated response is going to be anything you see in this red or this orange. So these helper T cells are reading this antigen on the outside of the macrophage and building this analysis protein, this cast or this form that the antigen is in, and they're going to release that interleukin 2. That interleukin 2 is going to cause the cytotoxic T cell to come over and read this analysis protein. So now our cytotoxic T cells have been activated. Cytotoxic, cyto, that prefix means what? Hopefully you said cell. And then toxic, when you think of something being toxic, you think of kind of like poison or bad for you. So this is going to produce some type of cell poison that's going to kill the cells that are already infected. They're going to read the MHC1 that's on infected cells and attack and kill those cells. So we could have a cytotoxic T cell that goes up here and reads this little antigen on the surface and beats up that cell. They can also analyze cancerous cells and the proteins that are coming out of cancerous cells and take care of those cells at the same time. They will kill them as well. Now you say, how does one cell in your body kill another cell in your body? Well, it's kind of neat. They produce a chemical called perforin. And this perforin is like chemical bullets, and these bullets are going to go poke holes in the membrane and cause that membrane to explode. And then the cell's gone. All the particles that get produced by these broken down cells will be eaten by the macrophage. Until the macrophage can get there and take care of them, though, those particles, all that cell debris, is actually the pus, okay, that you see. You all know about pus. Pus is a really gross word. But if you've ever had a pimple, you've been exposed to pus before. It's a tiny little infection that's caused inside one of the pores in your face. And as those cells get broken down and beaten up by the cytotoxic T cells, the ones that got infected by whatever bacterial infection was there, the debris that's left behind is the pus that comes out when you squash a pimple. So kind of gross stuff. Also, along with the cytotoxic T cells being activated, we also get these natural killer cells. These natural killer cells are going to kill according to what the antibodies tag. So just like we said a few minutes ago, those antibodies are going to be like little flags for the bad cells, the bad pathogens that are present in our body when we're undergoing an infection. So that flag is going to get picked up by the natural killer cells. It's going to attach to the cell and release chemicals that will cause that cell membrane to burst, and those cells will then be deemed inactive. All right, so let's go back and review one last time. all the way back up at the beginning. Okay, so we have a pathogen that's present in our system. It could run into an infected cell. What is the infected cell going to do with its last dying breath? That is right. It's going to post the MHC1 on its surface. The antigen that pathogen carried in will be posted on the surface. It's also going to release a chemical that will help tell other cells in the surrounding area not to come near it. That's right. The interferon will work by negative chemotaxis to warn the cells around it. Another idea that could happen is if the pathogen comes around and runs into a big white blood cell. What is that big white blood cell called? A macrophage. Very good. That's the big eater. And the macrophage is really proud of the fact that it could eat up this pathogen, and so it's going to do something with the pathogen in its body. That's right. The MHC2. It's going to post that antigen on its surface so that everybody else can know what to look for. Now, just a bunch of macrophages running around eating people is not good enough. We've got to mount an offensive. This pathogen is constantly replicating and causing us trouble. Runny noses, sneezing, all that stuff. So what do we do in order to activate a whole army of cells to get to work? Hopefully you said something about IL-1. We're going to release that chemical to work by positive chemotaxis to send in our reinforcements. And who is our reinforcement? That's correct, our helper T cell. So our helper T cell is going to work by positive chemotaxis. It's going to be pulled in by that IL-1 being secreted by that macrophage. And what is that helper T cell going to do? Right, it's going to analyze that MHC2, that antigen presented on the outside of that macrophage. And what is it going to do in return? It is going to release IL-2. Very good. Hopefully you're talking back to me so I don't just sound like a weirdo on this recording. All right, so IL-2 will be released, and we have two pathways set in motion. What are the names of those two pathways? Let's see if you can answer out loud. All right, the first one is humoral, and the other one is cell-mediated. Very good. All right, so let's talk about humoral first. The humors of our body are what part of our body? Good, the fluids of our body. In our fluids, we have B cells that are present. These B cells are going to analyze the protein, the analysis protein that was on the helper T cell to get an idea of what that antigen looks like. Through positive chemotaxis, it'll be drawn towards that helper T so it can read that little protein. And this is going to trigger maturation. What two cells is this B cell going to mature into? Well, automatically it'll mature into plasma cells. These plasma cells are responsible for building those antibodies, and those antibodies are going to go be little toddlers in our fluids and attach to all those little virus particles or all those little bacterial cells and flag them for us so we know what to pick up later. What's the other type of cell that's produced by a B cell? Good. Hopefully you said memory cell. Where do the memory cells stay while we're waiting to use them when we were waiting to get exposed again? Hopefully you remembered the lymph nodes. And then once we're exposed that second time, the memory cells will start producing those antibodies from the actual blueprint we got at the beginning. All right, so there's our fluids are cleared, but we've got some cells that were infected in the meantime while we were collecting all those pathogens. So the helper T cell, through the release of IL-2, will also activate cytotoxic T cells. These cytotoxic T cells are going to read that MHC1 on the infected cell and know that it needs to be disposed of. But how in the world do we get this cell to attack a cell of our own body? What is that chemical it's going to use? Good. Hopefully you said perforin. Perforin is like those little chemical bullets that deem that cell useless and uninhabitable. It's going to poke holes in the membrane and cause it to explode. We also are going to activate natural killer cells. They're going to read the antibodies flags that they place on those pathogens and take care of business there, taking care of all those antibodies that were labeling those pathogens for us for destruction. All right, so big picture, I think it makes a whole lot more sense now. If we go back to our cast of characters, you can see where everybody played a part, very important parts of our immune system. Without them, you would suffer from multiple different infections over and over in life. I've had mono once and I don't want it again. It was the worst sick I've ever been. So hopefully that makes a whole lot more sense now. If you have any questions, we will have a review session Monday afternoon in my classroom at 3:05 and Tuesday morning at 7 a.m. Hopefully this was helpful, not to steal the catchphrase from Bozeman Biology, but I hope that was helpful.